Everybody, I just promoted Griffin to panelist. I'm going to do the same for Kate and Avinash. Good morning, everybody. Good to see you here. 9 o'clock in these 24 hours. Super exciting panel coming up on using the vasculature to see if we can get a reference system, a latitude longitude system for the healthy human body, and not just one body, but hopefully male and female bodies. And so I will give it over to Griffin. And thanks so much for joining us on this 24-hour marathon here.
Thank you so much. Let me share my screen. Today we're talking about the healthy human body, but I have 3 little kids, and everyone with little kids right now is a little bit sick, so I apologize in advance. Thank you for the organizers of this event, including Kadi, for inviting us today. I'm Griffin Weber. I'm an associate professor in biomedical informatics at Harvard Medical School. I'm joined by Kate Castillo from Indiana University and Avinash Boppana from Revala. And I may add a little bit to their introductions as we get to their part of the presentations. Also like to thank Zarina Gauss. I think she's on the phone today. She's from the National Institute of Health, and was really the one that came up with the ideas that we're going to be talking about today. And myself and the group I'm working with have been expanding it and trying to make this a reality, as well as Kadi Bonner and many others for contributing to this work. So I'm going to start out giving an overview of the idea of a vasculature coordinate system of the human body. So we're presented with a problem, which is the creating map of all the cells in the human body. And it's a daunting problem. There's 37 trillion cells approximately in the body. Every person's a different shape and size. So how do we even go about thinking about how to create an address or a location of each of these individual cells? So we started by thinking about an analogy. What if instead of creating a map of all the cells in the human body, we created a map of all the buildings in a country? So this is a really interesting image that I found. What they did was this group took a satellite image of the United States and had a machine learning algorithm figure out from those images what's the rooftops of buildings. And everywhere where there was a building, they put a small black dot. And then this map here shows you a map of where all the buildings are. It's similar to a histology image where you can see a picture of all the cells that are stained. But this is just this overall picture is very far from being able to describe each of these individual buildings, the properties and where they're located. So how do we take an image like this of buildings or an image like this of cells? And how do we describe where each building is located on this map? So this is a house in Boston that I visit very frequently. One way of describing the location of this building is using the standard latitude and longitude coordinate system. So I can, if someone is telling me the address of this building, so I can go visit, they can say it's at 42.302708 latitude and negative 781.0 longitude. So obviously people don't describe locations of buildings this way. Even though it's a natural way of describing coordinates, it's using the standard latitude and longitudes. It comes out of GPS and other things. So what do we use instead of that? We use something like this. This is 49 Oldfield Road, Boston, Massachusetts, 02121. So we're actually using a road-based coordinate system to describe the position of the house. This is a street and telling you where along that street this building is located. It's not really the location of the building, but we're using the road that's adjacent to this building to describe its location. This is telling you the general region of it, and then this is the specific road. And then that road has one end where it starts at a number 1 and it goes to the other end where it's about a 60. And this is telling you where along that length this particular building is located. So why are roads a good coordinate system for locating buildings? And why might we use blood vessels as a good coordinate system for locating cells? And I'm going to give some parallels between these two things and give an argument that explains why we think the blood vasculature can serve as an addressing system or a location system for cells in the same way that we use roads in daily practice for describing location of buildings. So first of all, it's the latitude and longitude is not very practical. For me, if I'm driving to this house, roads get people to buildings. So the road address is giving me instructions on how I can get to that building. Same way vessels get oxygen to all living cells. So these are the pathways that you use to get back and forth to these objects. As a result, most buildings are near roads. If they're not near a road, it's hard to get people to those buildings and to get people out of them. Same thing, living cells are going to be near blood vessels. Roads cross the entire country. When I'm at that building in Boston, I can drive all the way to California without having to lift the car up. It's one continuous highway system that can cross the entire country, going from big highways all the way down to small alleys and driveways. Same thing, the blood vessels span the entire human body. It's one closed endothelial system. Otherwise, the blood would leak out. So it starts at the heart, and the pathways lead to all different parts of the body. Roads, I said, scale from interstate highways all the way down to the smallest driveways leading into a house. And blood vessels scale from major arteries all the way down to capillaries. If you're going to build a new building, it's helpful to first construct the road and then build the buildings around that road. And similarly, organs often develop around new blood vessels. First, the blood vessels form, and then other types of tissue can form around them. And finally, you have road signs that tell you where you are located on the map. And our hypothesis is that various biomarkers, such as endothelial cell genes, microvascular patterns, and others, can help you describe the position of vessels in the body, and then you could describe the location of cells relative to those vessels. So how do you start building a map of roads? So the first thing you may want to do is build out the map of the highway systems. So this is another interesting image I found. This is a map of all the highways, but the width of the highway in this picture is related to the number of long-haul trucks that pass over those roads per day. So the thickest highways over here are the ones that have the greatest traffic. And if you remove the map of the US here, you might look at this and think this is an actual arterial blood system. There's a lot of parallels here. One thing that's interesting, though, if you're building a map of buildings, when you look at these interstate highways, a photo of them, there are no buildings around it. If you're trying to come up with a map of pictures of buildings, you need the smaller streets. But in order to link all those streets together, you need these highways. In the same way, you need to start with the major blood vessels. We start with the aorta and other major vessels in the body. And that's the sort of the framework that we use to then link on the smaller vessels. Same way as you build out the highways, and then that allows you to create smaller roads that branch off of them. If we're starting from scratch and there's no street signs, can we still determine location of where we are based on the patterns of streets? In this group over here, what they did was they took different cities, both in the US and across different countries, and they looked at different types of roads, avenues, drives, boulevards, and others. And what these circles show are the directions on a compass, north, south, east, west of those roads. So in Atlanta, Georgia, for example, the avenues move east and west, and the streets move north and south. Each city has a distinctive pattern. So if you had a map of the lines of the different roads in a city, and you're able to identify which ones were avenues, streets, or others, it gives you a distinctive fingerprint that you can then use to figure out where you're located. Similarly, there's different types of blood vessels that you can that have different kinds of lengths, sizes, react to different stains. Can we use the different types of vessels based on arteries, veins, or even subtypes of those based on gene expression, as well as their geometric properties such as length, diameter, branching patterns, to uniquely be able to describe where we are in the body? Without a sign naming each individual road, it's hard to give a unique address to every single building. But maybe instead of looking for every single building, we're looking at categories of buildings and how they're related to categories of roads. So this was a classification of buildings from 1932 that was used for city planning. They're saying, give us different kinds of roads and different kinds of buildings, and we draw out maps for those and help design an urban environment. So here it's they come up with a coordinate system, number of families per house, and the number of shared walls. And then these are different categories. They name them, and they can plot those on maps. And similarly, in a vascular coordinate system, can we describe different kinds of tissues and cells that are near distinctive types and patterns of blood vessels? So here's a map of Boston. Boston has a long history of different neighborhoods being developed, and different roads in different neighborhoods in Boston have distinctive patterns to them. For example, this is the Back Bay region, where there's a very grid-like pattern of roads. This is one of the more recent additions to Boston. It tends to remind me of skeletal muscle vessel patterns. There are these row houses located on these streets. It's a very distinctive neighborhood in the Boston area. Here in Beacon Hill, there are these cobblestone roads that kind of remind me of the branching patterns of vessels in the brain. You know, there's neurons and other cell types adjacent to these vessels. And then this is the older part of Boston where the streets are a bit chaotic. That kind of reminds me of the vasculature and the spleen. Here we have a mix of new buildings and some of the older ones that are still remnants of Boston's past. These different neighborhoods are connected by larger highway systems. Interestingly, I-93 is sometimes nicknamed the central artery. So they even use this biological references sometimes to describe roads. There are grid-like streets in Boston, but there are also grid-like streets in Manhattan, and the types of buildings next to those streets are very different. So just knowing that microvascular pattern may not be sufficient, having the name of a larger vessel nearby might be important too. So if you're saying a grid-like street near I-90, you know it's this kind of building, and if it's a grid-like pattern near one of the highways in New York City, then you're going to get these types of high-rises. So putting this together, if we're going to create a vasculature common coordinate framework or a kind of coordinate system for the body based on blood vessels, we can imagine the body not anatomical, but sort of drawn out in a different way with the heart in the center, with different blood vessels reaching out to all the different organs in the body. Main vessels like the renal artery indicate the macro scale region of the body, just like an interstate highway tells you what state or region in the United States that you're in. And then the microvascular geometry and biomarkers give you distinctive patterns or neighborhoods on the micro scale. So once you know that you're on the renal artery, once you get down to the capillaries, if you have a cell that's adjacent to a glomerular capillary, you have a good idea where you are in the body. And then a cell's address is a named vessel plus the cell's distance to that vessel in a specified microvascular neighborhood. So for example, if I'm looking in a histology image, I might say that this cell is 20 microns away from a glomerular capillary downstream from the left renal artery. And that doesn't tell me exactly where I am because there's going to be lots of glomerular capillaries, but that might not be so important. Just knowing that this is a cell that's a certain distance from a category of blood vessels, that same pattern is repeated many, many times, and it gives me a sense of the different kinds of tissues, where they're located, and their relative position to each other. Here I'm looking at one of these vasculature loops. So you're starting with the renal artery, going all the way down to capillaries, and then back out through the renal vein. In this vascular coordinate system, I don't need to identify every single one of these loops. There could be millions of them. I just need a typical one. So for example, if I'm describing the types of buildings in the Back Bay region of Boston, I show one example of those streets. I don't need to give every single one of them. So here I'm taking one example loop, I'm pulling it out, I straighten it up, and I can show along this path, going from the arteries out to the veins, what's the different kinds of cell types that are surrounding those vessels, and how does that pattern change as I'm entering and leaving the organ. So I first described so the rationale for this and what we're trying to accomplish. So what we've been working on the last couple of years is constructing this database of blood vessels. So the road atlas has the list of all the streets and where they're located, and similarly, we're trying to come up with a blood vessel atlas, a database that gives you as much information as possible about blood vessels, so you can use that to describe location of individual cells. A starting point for this was a standard ontology called Uberon. This is a list of thousands of anatomical structures and relationships between them. Uberon, I mentioned, had thousands of blood vessels and other kind of anatomical structures in many different species, not just humans. We pulled out 747 individual human vessels out of Uberon. This circle graph over here, each one of the dots represents a different one of these vessels. The ones in pink are vessels that are connected to other vessels through branching relationships. So many of the vessels said, yeah, this vessel here branches off of this vessel over here. But a lot of them are just individual vessels. They mention the vessel, but they don't describe how it's connected up. We need to connect all these things up in the same way that you want to see how roads connect to other roads. We want all the vessels connected up. So we did this in a pairwise fashion. We took two vessels at a time. We tried to find the connections between them and draw those things out. And eventually we end up connecting all those vessels into 4 trees or branches. One branch of trees coming out from each of the different atriums and ventricles, chambers of the heart. So we have a left ventricle, arterial vessel tree, right atrium, left atrium, and right ventricle. So it's color-coded over here. So we start with all the Uberon vessels and link them together. And then we work with domain experts to fill these in. Uberon wasn't complete. There's some duplicates and other things, but this was a good starting point for us. Visualizations are very helpful. We had some just simple ways of looking at these blood vessel trees in a more anatomical position. And this just helped us make some quick checks. Did we connect a brain blood vessel to the pelvis, for example, and we have to make those corrections. It's hard when you're looking at a database of hundreds of structures to check and make sure it's okay. When you're visualizing things, it's much easier to think about if this is correct, because our domain experts are usually visualizing these things in an anatomical way, not in a database or spreadsheet way. We did a number of simplifications. If you try to create a database of all blood vessels, there's going to be millions and millions of these. So to try to simplify things, we collapse things where we can. So for example, the abdominal aorta branches off to a left renal artery and a right renal artery. So what we say is, instead of showing all the branches of each one of these individual renal arteries, let's come up with what we call a virtual vessel, something called renal artery. There isn't a real vessel called renal artery. It's either the left or the right. But we put renal artery in the database and indicate that this is just a virtual made-up vessel. And then we show all the branches off of that. So there are a bunch of segmental renal arteries that branch off of our virtual renal artery. And then we can combine these segmental arteries into another virtual vessel called segmental renal artery, and so on. So we can describe the different branching levels in the kidney without having to explode the database exponentially with each of the individual vessels that are on it. One of the challenges we will have moving forward on this are variants. There are general patterns that are found in most people, but there are a lot of variations across the population. This is variations of the aortic arch. There are different classification systems for it. So this is one naming system for those branches, and this is a different naming system. So we have to go through literature and figure out which is the one we're going to use for this reference atlas. It isn't just in the aortic arch. All through the body, there are these known variants and patterns, and we'll eventually have to incorporate this into our atlas. As we start getting into the microvasculature, cell types and biomarkers become more and more important. There's a lot of known vessels, and the Human Biomolecular Atlas Project and other consortia that we're working with are doing a lot of work right now to try to get better understanding of the different biomarkers that occur throughout the blood vasculature and how these differ between different organs and different parts of organs. There is a number of people who help construct this database, and then we have domain expert reviewers who validate it. We have 3 primary reviewers: Mark Valushka, who's a vascular pathologist; Rajiv Malhotra, who's a vascular biologist; and Sujin Lee, who's a vascular surgeon. It was important to get people from these different domains because in different fields, people view the blood vessels differently. In one field, there might be a single vessel where people call it one vessel, and in another context, people view the different segments as individual vessels. There's different naming conventions for blood vessels. So having people from different backgrounds gave us some of this variability on how people describe blood vessels. Sometimes it was a challenge taking the comments from these 3 different reviewers and trying to deal with the differences between them and come up with a consensus for our database. Some of the challenges we had as cleaning up the data are misspellings, both in what we did and some of the source system, standardizing names. Some people have names of vessels that have dashes in it. Sometimes they don't have the dashes. There are multiple names for vessels, so you can end up with duplicates. I talked about paired vessels like left and right renal artery that we had to merge to simplify the database. We're trying to develop this atlas as more of a tree. So a large vessel branching off to smaller vessels, but the vasculature isn't purely a tree. There's lots of loops. Blood can flow in different directions. You have to handle all those special cases. And then we want to link to other databases. We're not the only database of blood vessels. I mentioned Uberon. There's also functional FMA, another ontology of anatomy, and then some historical databases that have been around for quite a while. We summarize this database in a preprint that's currently in review. We currently have over 1,000 blood vessels in this database, along with 10 cell types, endothelial cells, fibroblasts, other things related to blood vessels, and 10 biomarkers. This is rapidly expanding. As you combine the biomarkers and cell types with the blood vessels, you get about 9,000 different combinations of vessels mapped to cell types, mapped to biomarkers. The database we have is not just a list of names of vessels and their connections. There's lots of additional data that we connect to it, such as the vessel type. Is an artery, arterial, capillary. For some vessel types like capillaries, there are subtypes. The mappings to different body parts and subparts of those organs where a vessel supplies or drains or passes through it. There are portal systems that we indicate, such as hepatic portal system or renal portal systems. Some vessels only occur in female or males. We have to indicate that. I mentioned challenges of anastomoses and variants that we have to indicate. We have those 4 different branches, the arterial branches, venous branches, and then pulmonary and systemic. We want to connect these up. So where does the artery ultimately go through a capillary and connect up to the vein? So we have those artery vein connections. The virtual vessels that get collapsed, and then a branching sequence, a large vessel like the aorta. There are many vessels that branch off of it, so we indicate this is the first vessel, the second vessel, and the third vessel. And all these things are used to help you, again, locate where the vessels are in the body. The same way that streets, you could have the street type, is it an avenue or is it a road? The orientation of it, is it a cobblestone road or a paved stone or road or a dirt road? So in the same way, we're capturing the more information or metadata we can have about these roads or the blood vessels, the more precise we can describe the location. So with that introduction, I'm going to switch over to Kate Castillo, who's going to talk a little bit about how we can learn more about the geometric properties of larger vessels. And then Avinash will take over and describe different approaches people are using to understand microvasculature.
Wonderful. Thank you, Griffin. Yeah, so I wanted to start by kind of going over a project that I did to see the extracted human circulatory system from a cadaveric donor, and then how we can obtain geometric properties in different methods. So both using the sort of dissection that I had completed, but then also looking at models and other methods that we can start to visualize geometry. So if you could go to the next slide, please. Okay, so here is some images from my capstone project. So this is for my master's degree. And this was, as far as I know, the first recorded extraction of the human circulatory system using just traditional anatomical cadaveric dissection techniques. So we didn't use plastination. We didn't use infusion with plastic. So you might have seen in one of the earlier slides, there was an image from Body Worlds where they injected all the vessels with plastic and then kind of removed the tissue. So this was actually going in and doing a dissection. So with this, we dissected about 400 arteries, 63 veins, and then the heart and liver in one piece. It took about 250 hours. I was the primary dissector. I did most of the dissection on my own. And I just want to point out some things that we can see in some of these images, and especially highlight some of the things that Griffin had already talked about. So there's this long image where you can see the full circulatory system, kind of a zoomed out view. And up at the top, you can see the arteries in the brain. You can see kind of these large vessels coming up and branching and then kind of curving in. That's the internal carotids. You can see the heart and the liver, which the liver had to be connected due to the portal system, right? So if we had taken the liver out, then we wouldn't have had that connection of the portal, the venous portal system, the hepatic portal system. And then you can see the arteries and some of the veins of the arms and the legs. So if you look at the arms and legs, there's kind of two parallel lines. One of those is arterial, one is venous. And then you can see there's a lot of blood vessels in the abdomen, in the intestines, in the pelvis. And so you can kind of get this general macro scale view of some of these structures and how they actually are arranged at a gross anatomical level. I do want to note in this kind of second image that shows about half the dissection, there is a distinct difference in color in different places. And this is just could be through embalming techniques, different properties of blood vessels, but there are definitely differences and changes. So if you look right across the heart's kind of right in the middle, and then on either side, there's a large kind of branching tree that has this dark pinkish brown sort of color. That's where the lungs were located. And then up the brain arterial structures are more kind of bluish looking. So it's just some interesting things that you can note. And then talking about some of these challenges that Griffin mentioned, such as variance and anastomoses, they can be very clearly highlighted. So this specific donor had an interesting variation in his abdomen where he didn't have a common hepatic artery that branched off. Rather, his left hepatic artery kind of came off of the left gastric artery. So one of the other branches of the celiac trunk, which is one of the main branches of the abdominal aorta. And then the right hepatic actually came off the superior mesenteric artery, which is the next branch down. So this is a pretty different arrangement, and that's very common, right? So we have to think about how to tackle that problem when planning this vascular common coordinate framework. And then another thing I think that's really beautifully highlighted in this dissection is anastomoses. So you can see a couple kind of at the bottom in those smaller images. There's the anastomosis in the brain, the circle of Willis, or the arterial cerebral circle, and this is kind of located at the base of the brain, and you have this beautiful actual physical circle that is this connection of these arteries. And then another one I think is lovely to see are these arterial connections at the tips of the fingers. So you can see where the hands are located, and there's a zoomed in image. You have digital arteries that then connect to each other. So, we can physically see these sort of things if you do a traditional sort of gross dissection, but then we want to think about what are other ways that we can start to think about this project. So if you could go to the next slide, please, Griffin. Okay, so we have this dissection. Currently, it's actually being plastinated in Toledo, Ohio. And once this plastination is complete, we can start to obtain certain vessel geometries. So you can start to get length, diameter. You can measure these physically just using calipers. And then I want to note that while we can measure some of these things, terminal vessels that just continue to branch and branch, we don't really know the accurate length because you can only trace so far when you're doing a gross dissection with, pretty bulky tools. Even if you have a nice pair of really sharp scissors, they're still pretty bulky. So you can really only get so far. And then we have, again, I kind of highlighted here the variations in the anastomoses, but I think we can start from this gross view and see these kind of larger structures. We can get some measurements. We can start to learn some information, but it's only one sample size. It's only one donor, right? And so what are some other ways that we can think about how do we get these vessel geometries? How do we understand the actual shape and morphology of these structures? So if you could go to the next slide. So one one way of doing this is to use 3D models. So there's this database, the Visible Human, that was basically a physically sliced human, and then those images were compiled to create this large database. And one of the tasks coming out of this is to create 3D models. So we have these 3D models that were developed of the vasculature. So this is the venous and arterial system. And you can take those 3D models, and Lu Shen, a student at Stony Brook, she took a lot of these structures and was able to automatically generate geometric properties. So she could go in and figure out the minimum and maximum and mean diameters and lengths. You also, there's some issues there where you might have some collapsed vessels, you might have some issues with the 3D models, depending on what kind of software you're using. But you still can get a good amount of information. So we have 210 vessels, 102 male, 108 female, that give us some geometrical properties, and that's really informative in many ways. But also, people have been studying anatomy and anatomical geometries for quite a while. There is a lot of literature out there that looks at different geometries, typically for larger structures, especially larger structures that have significant clinical implications. So for example, the differences in abdominal aortic diameter can be very important for concerns of aneurysm, things like that. And so we have these literature review studies that study individuals or populations. There's different methods of looking at this. You can use CT scans, ultrasounds, you can use a cadaveric study. So there's many kind of ways that people have looked at these geometries. And really one of the goals of this VCCF is to take some of this information and pull it together so we can augment what we know and how we understand the way the vasculature allows us to navigate through the human body, not just of one person, but across multiple different populations and demographics. So if you could go to the next slide, please. So one thing that I think is another kind of way that we've thought about doing this is, how can we use machine learning and how can we use some of the advancements in technology to further this effort? And so I want to talk a little bit about a Kaggle competition. So Kaggle competitions are a competition that's hosted by Google. It's one of the largest machine learning platforms. And what it does is it poses a problem to the general global community and allows the global community to collaborate to generate solutions. So algorithmic solutions to the question and the issue presented. So through HubMap, they have used Kaggle previously to also help answer other questions that have been brought up. So one of these is to talk about identifying different structures in histology slides. So the first Kaggle competition was about identifying functional tissue units, or FTUs, which include the glomeruli in kidney histology slides. So is there a way we can automatically annotate these things without having to go in and have a pathologist or go in and have a histologist manually draw these structures? And then the second Kaggle competition identified other kind of cellular structures, not just in kidney, but in histology slides from 5 different organs. So how does this relate to the VCCF? If you could go to the next slide, please. The third and fourth competition, and Kari Borner has really been a wonderful force for this, is we are devoting the third and fourth Kaggle competitions to the vascular common coordinate framework. So the third is looking at 2D blood vessel segmentation. So instead of trying to segment out structures that might have like FTUs, functional tissue units, specifically in the kidney, what we want to look is the blood vessel, which of course has lots of functional purposes in every organ. And so what we want to do is create a machine learning algorithm that can segment out capillaries and arterioles and venules. Of course, there's some challenges there, but I think, you can see some of these vessels in these slides. And so can we automatically segment them out? And then the fourth Kaggle competition is going to be looking at 3D blood vessel segmentation. So this is using HIP-CT data. And I believe Peter Lee was previously on the call and presenting at the last session. And so we're collaborating to use HIP-CT data to look at 3D structures, and specifically arterial 3D structures. And that's going to be at a slightly larger scale. But once we have those 3D models, then we can start to run some of those geometrical studies, such as those done by Lu Shen, to gain more knowledge and information. And so if you want to go to the next slide, this is just an example of what we can do for the CAGL3. So you can see this kidney histology slide. There are some blood vessels that you can see here in the yellow. Those are highlighting capillary/venule structure. Renal pathologists have indicated that it's very difficult to distinguish between the two due to physiological function. And then there's also kind of a larger vein, possibly structure, and an artery and some arterial structures. And so, we can see, even in this image, you can see some red blood cells that are tracing through these capillary and venous structures that kind of are a giveaway, even if you don't have immunohistological staining or these other sort of properties. Of course, that data is also useful to help augment and tell us about some of these structures. So, this is just to highlight some different methods that we can learn, and especially learning about microvasculature. Once we get past what you can see from the gross level, once you start to get down to, almost single cell resolution, it becomes, there's a lot of blood vessels. And so how do we tackle this big challenge, right? And then the CAGL4 is kind of still in process, but that, again, we'll look at 3D segmentation and hopefully give us some more geometrical properties there. Yeah, thank you very much. And I think Avinash is going to be talking about the methods for generating a vascular fingerprint. Thank you.
Thank you, Kate. So kind of just to summarize, Griffin and Katie, highlighted how we computerize the macro and semi-micro scales of this vascular coordinate system. So this is kind of like the highways and roads of our analogy. And they use this, they can do this by using well-defined connections between known blood vessels in literature or cadavers, for example. So the next few slides, I'll be focusing on the microscale or the neighborhoods of our coordinate system. And I'll briefly detail 11 examples from literature that motivate how we can create these vascular fingerprints. So these would be quantitative signatures of the various neighborhoods of the microvasculature that interact directly with all the cells in the human body. Next slide, please. So as you get to the micro scale, things start to become generic and indistinguishable. There are billions of capillaries, venules, and arterioles. So what we can do in this regime instead is quantitatively capture the state of the system. And the system of interest are these region-specific neighborhoods of the microvasculature. So we dived into the literature with a guiding question: what can we measure about the microvasculature that can serve as a fingerprint? And we identified 3 categories and methods. The first one was vascular topology and morphology, or how imageable blood vessels are shaped and structured. 2 is omic measurements of vascular cells. So this involves measuring expression levels of biomarkers such as genes in cells that the blood vessels are composed of, like endothelial cells. And finally, 3, we have flow and gradients of fluids and chemicals. So how do substances physically move and distribute within these vessels? And the idea is to stitch together a variety of these methods to increase the precision of our fingerprints, where different high-resolution data sources will give us a clear picture of the differences between various neighborhoods of the microvasculature. Next slide, please. So I'll kind of start to dive into some of these papers. So this paper shows injected fluorescent microspheres throughout the microvasculature of 4 different tissue types. So you have skeletal muscle, brain, kidney, and spleen. It's really indicating how does the inner lining of these vessels, how are the inner lining of these vessels structured? So we see elongated and continuous streaks of green in the muscle and brain tissue. And this corresponds to an inner lining of these vessels with minimal gaps. It's very smooth. However, in the kidney and spleen samples, the microspheres don't generate that pattern. There's a lot of pores and perforations. It's indicative of the function of these tissue types. The key takeaway is that the structural properties of the microvasculature in different tissue types have corresponding signatures. Next slide, please. So this paper focuses on quantifying the microvascular architecture in mammary carcinoma, specifically in rat ovary tissue. And it's motivated by the fact that cancer causes abnormal blood vessel growth. So here we can see different quantitative distributions of topical and morphological metrics, including capillary diameter, branching order, and intercapillary distance. And the focus on this paper is on diseased tissue, while the vascular coordinate system is focused on healthy tissue, but it provides some motivation to investigate how can we apply this metric-based framework to healthy tissue. Next slide, please. So this paper is a lot more different, and similarly, it's looking at diseased tissue in the human breast. The key differences here are the authors have a computer automated approach to quantifying various metrics of the microvasculature, and they also have a comparison sample to benign tissue. So they image the microvasculature using this ultrasound Doppler imaging, which is a sound wave-based approach to imaging how blood flows throughout these vessels. And then they develop a method to quantitatively analyze this. So in particular, this figure has an emphasis on vascular tortuosity, or how these vessels are curved. Figures J and K show two of the metrics that they developed. One is sum of all angles, and another one is a arc length based distance metric. And the key trend to see here is different distributions, one in red and one in blue. We see differences in the distributions of these metrics between malign and benign tissues. Although this is a disease focus, again, it's another category of the types of properties we can measure. And in this case, it's how the vasculature is curved. Next slide, please. So this paper uses whole brain MRA imaging of the human brain to show age-wise differences in topological metrics. So the scatter plots on the right are showing differences in these well-defined metrics, tortuosity, fractal complexity of the vessels, and branching length. And we start to see some trends as the age of the tissue increases. So, the key takeaway here is different biological states of the tissue, whether it's age, maybe it's sex, maybe it's location, have similar dissimilarities in their topological metrics. Next slide, please. So the next few papers I'm going to focus on are omic-based biomarkers of the vascular cells that are composed in the blood vessels. So these can be endothelial cells of the inner lining, smooth muscle cells, etc. And this paper specifically focuses on the brain tissue of the mouse. So the authors use a clustering approach to characterize cell subpopulations of brain tissue, and then they attempt to find their gene markers. So the figure on the left is showing cell type specific markers of genes for various vascular subpopulations. And by subpopulations, we're not just looking at smooth muscle cells or endothelial cells. We're diving in one level further. We're looking at the arterial, venous, and capillary subpopulations of each of these cell types. So each of these colored spikes represents the expression level of that gene in a single cell. And the key takeaway is that even in a single tissue cell type, different subpopulations of the same cell type have distinct gene markers. And the figure on the right goes one step further, revealing the zonation properties of endothelial cells along this axis they've defined, the arteriovenous axis, where you go from the venous part of a microvasculature network to the capillary part to the arterial part. And the spikes, again, are corresponding to expression levels of the labeled gene for a single cell. And the interesting property to note, there are gradual increases and decreases in expression level as you traverse this spatially motivated axis. Next slide, please. This is another paper that again focuses on finding gene markers of vascular cell populations. And here they're specifically focusing on endothelial cells in the mouse lymph node tissue. So the figure shows different endothelial cell populations and their corresponding gene markers on the top, while the heat map portion of the figure shows the mean expression level of these marker genes in each of the endothelial subpopulations on the left. So it's kind of a pairwise comparison of marker genes along all these different subpopulations. And the interesting trend to note is the diagonal streak of high expression. This indicates we have gene markers that can differentiate between subpopulations of these endothelial cells in this specific tissue type. Next slide, please. So this next paper is especially interesting from a spatial perspective, and it focuses on endothelial cells in the mouse liver. This paper develops a method to infer trends in these endothelial gene markers as you traverse through zones of the functional tissue unit of the liver, specifically the liver lobule. So each liver lobule has a relatively consistent geometry with a central vein to portal vessel axis. The central vein, being in the center of these discs, and as you traverse outwards from these liver lobule discs, you get to the other end of the axis. And the authors split this axis into 7 layers. So the figure on the left is showing how specific gene markers have positive or negative expression changes as we traverse the spatially motivated axis in the liver lobule. Next slide, please. So while the previous omic papers we talked about were focused on gene markers between vascular cells in the same tissue, this paper and the next one are focusing on gene markers comparing the same vascular cell between different types of tissues. and the specific vascular cell they're focusing on are endothelial cells. So here we have a heat map of microarray gene expression data for 3 RNA samples from each of the organs listed. So we have brain, heart, and kidney, and the mini columns within each organ represent the replicates per organ. The y-axis is measuring gene expression of around 3,000 genes. And again, we see these color-coded blocks. And the key takeaway is there's some evidence that endothelial cells in different tissues have discriminative gene markers. Next slide, please. So this paper is much more recent, using more advanced technology, and it does what the previous paper did on a much larger scale. So you have many more organs, and it's also using single cell sequencing instead of bulk sequencing. And again, here they're showing the expression of marker genes on the x-axis and a variety of mouse tissues indicated on the y-axis. Again, more genomic-based evidence that endothelial cells have tissue-specific biomarkers throughout the human body. Next slide, please. So the last 2 papers are more unique instances of technology, so I'll kind of dive in a little deeper. Here we have an epigenomic technique. So where instead of measuring the expression level of genes transcribed from the genome, we're measuring patterns of the chemical tags on the genome itself. So the specific assay being used is something called ATAC-seq, and it measures this property called chromatin accessibility of the various regions in the genome. So very simply, greater accessibility or open chromatin means that regulatory elements like transcription factors can bind to these regions and regulate gene function, while closed chromatin usually prevents that. So this figure specifically measures the general distribution of this property, chromatin accessibility, near differentially expressed genes between endothelial cells in brain, lung, liver, and kidney tissue. And essentially each point in the scatter plot is measuring the regulatory potential of the same region between two different tissue types, where the color code represents that region was part of a marker gene for the x-axis tissue or the y-axis tissue. The key trend to notice is that the correlation between the 2 brain replicates in the bottom right panel is much stronger than the correlation between the panels with different tissues. So it provides some evidence that epigenomic profiling as well can profile differences of vascular cells between different tissue types. Next slide, please. So this last paper that I'll talk about in this presentation focuses on a biophysical marker, specifically the field-wise flow of blood throughout the vessels of the microvasculature. And they focus on liver and kidney samples from the mouse. So the authors are using this image processing method to produce images called chymographs. And essentially they quantify capillary blood flow over time, their velocities within a single static image. So the figures on the left are showing a symbolic representation of what they're measuring. You have different segments of vessels with different velocities of the blood flowing through it. And on the right, we see the extracted metrics they're developing, like velocity distribution of these different segments in the microvasculature, as well as how velocity changes over time, how flow is changing over time for specific segments. And I think the key trend to note, I've quoted the authors, but they were actually able to notice differences in flow rates between various regions of the liver. Next slide, please. So I just want to summarize real quick. A lot of these methods profile different aspects of the microvasculature, whether it's branching patterns of vessels, expression of biomarkers in vascular cells, or blood flow within these vessels. So the common attributes were that quantitative metrics can be extracted, and they present differently based on a comparison setting. So these settings were healthy versus disease, different tissue types, different cell types, or different locations and spatial axes. So our eventual aim is to identify and validate the methods that can be applied to healthy adult human tissue in the comparison setting of microvasculature neighborhoods. So this will be kind of the next step in fully realizing a vascular coordinate system. Thank you.
Thank you, Avinash and Kate, for your excellent presentations. I see we have a number of questions here. So let me pull up our list. Okay, the first one was, what role might lymph vasculature and the peripheral nervous system play in the latitude, longitude coordinate system for the human body? So we're talking about today our role in pulling together the blood vasculature. We didn't mention that there's parallel efforts right now to do similar databases of the lymphatic system as well as the peripheral nervous system. So in the end, all of those combined might be important to help to triangulate between, you have arterial distance that we're talking about today, how far are you from the lymphatics and how far are you from a nerve, and that might give you better precision and location than the arterial system as well. And then we have Zarina Gallus, who's joined us here today, and she can fill in any additional things she'd like to add. She's the person who first sort of came up with this idea, and then we've helped expand it out over the last couple of years.
Thank you so much. So first, I wanted to congratulate all of you. Amazing amount of work. We are getting really closer to what we hope we will have in the end. So just to add a little bit to your comment for this question, I wanted to say that probably being able to map nerves and lymphatic systems will also add a lot to mapping function. Because they are involved in specific functions. So if you go from that sort of big picture level to deeper, the mesoscale and then microscale function becomes much more localized. And I think nerves may not get as close to every cell in the body as the blood vessels get. However, they are going to be very deeply involved in creating the function for every, even like a microscale, and then going up back to the macro and full body. That's all. We don't have a lot of time, but a lot of interesting questions, I think.
Yeah. Our next question is very interesting one that Katherine might actually want to join in on. What is the exact difference between an atlas and a coordinate system? So first, my interpretation of this, with an atlas, you're using a coordinate system to describe relative connections between different things. So with an atlas, you need a coordinate system. The coordinate system is the different axes or properties that describe where you are. What we're doing here is a combination of both. Our blood vasculature database, in some ways, is an atlas of blood vessels. And the coordinate system is internal. It's the connections between vessels. So when we have renal artery, to describe the location of that, we're saying it's branching from the aorta. So the branching relationships is sort of an internal coordinate system that's telling us how the vessels connect. And then once we have that, the blood vessel database we have is then being used as a coordinate system for other aspects of the human reference atlas. Kadi, do you want to add to that at all?
Good morning, everybody. Good to see you. Great presentation. I was really, really interested also in what Avinash just presented, and then another question on this one. The background for my question about Atlas and reference systems is really about how to define those two. And I think there are different definitions going on. Maybe if you ask the different experts to tell exactly what they think azimuth reference is or the new long map reference, they would actually have quite different answers. And in order to communicate this properly to the world and to make progress, I think we have to agree on de facto definitions, which of course can always be improved. And so I wanted to learn from your end, because you had time to focus on the vasculature CCF, exactly how you would define it. Mm-hmm. And maybe that's homework. Maybe you can't do this in front of the camera right now.
Yeah, I kind of gave a simplified answer, but yeah, these are very, that's an excellent question here. And as you said, there's lots of interpretations.
I can throw in one quick opinion. So once we have the atlas of the vasculature itself, so this is what you are seeing being developed under your eyes. So you start from the whole body level, and again, you go all the way to the single cell level capillaries. So that's the atlas or the map of the vasculature. but then that can be used to map all the other cells based on their locations, either interactions, direct interaction, or being within the watershed of a specific vascular structure. So that is the reference that we are talking about. So that's the vascular CCF. So can you tell where a single cell of the liver is located in connection to an endothelial cell of the liver? And I just want to expand a little bit of what was presented and put together maybe a little bit the whole concept. One is having a roadmap on how you get to every cell. And the other one, a little bit like Avinash was presenting, is to be able to print not only the pattern of the vasculature, but the actual expression of genes in every endothelial cell, the smooth muscle cells, other vascular cells, as well as the cells next to them. So that's more like a GPS. So rather than telling you take this street, take the other street, I just give you a GPS and you know exactly where you're supposed to land. So I don't know if that answers, but I tried.
We have it on record now, Zarina, and Mark Kalushka just gave another definition. To him, a tissue atlas describes the different pieces, but the framework connects all of the pieces and, importantly, identifies where multiple copies of a vasculature segment exist. And so maybe that leads over to another question I had for Avinash or everybody here on the call, really. How big of a tissue block or section or suspension do you need in order to know where it came from? Do you know yet? And exactly what assay types do you need to run on it, right? Because that's really where many people are asking questions. How much do you need? And as Zarina has been arguing, well, a pathologist can look at a piece of tissue and they would know it comes from that organ. And maybe not exactly where from the kidney cortex, right?
But you can, because I think that what you get is the phenotype of the cells, the pattern, the organization. So if you look at composition, the type of cells that are there, the pattern, I think Mark would say the same thing. And I'm going to just guess that probably a few hundred cells just for the visual inspection, if you see everything in context with no specific stains. Now, when you come to single cell assays, you need enough cells to actually separate them in batches and things like that. And if you have histology with stains and everything, maybe you need even fewer cells to tell where you are.
Yeah, just to add on to that, I guess, for sequencing studies, especially single cells, sometimes it ranges on like 50,000 cells up to like even 1 million cells. But I think the key thing is being intentional about the sequencing study. So like if the goal is to find a gene marker that has a global, difference between different tissue types, then you need to also have the study measure those different tissue types. That can, expand like the size of the tissue that needs to be processed.
It's going to be really important to figure out what are these units that repeat. It's just not going to be practical to come up with a database of every individual vessel and body. We're going to have to come up with patterns and show how those patterns repeat and how those become larger patterns that repeat. So we're kind of describing it as one tree right now, but it's finding what these units are and is really going to be essential to making this practical.
Excellent. There are some more questions in the chat. Feel free to answer them via chat. I would like to thank you one more time. It's wonderful to have this element of the CCF work covered in the 24-hour session. And please continue your good work. I'm a true believer in that this might actually work and help us cross scales in the human body, which is really an enormous challenge, given how many scales there are, and also to help us get to a reference system that many can use efficiently. I think it has to be easy to use, just like a GPS system. But of course, the technology behind GPS is not trivial to get right. And as you know, there were many technological advances that were needed to get us a latitude longitude system, including having a watch, a clock that you could use. Otherwise, you couldn't have latitude longitude measurements. So I think similarly here, we have many new technologies come into into existence. Also, some of them presented here, and I still see Peter on the call, which is wonderful. But ultimately, I think we all together can get us a reference system that works for something as complex as our own very own bodies. Thank you.